Okay, so our next speaker is Lei Zhang from Peking University, who will tell us about noise attenuation during the development of spatial patterns.
Thank you. Thank you, organizers' invitation. Great to be here. It's my first time to Cambridge. Very nice place. So my name is Lei Zhang. I'm from Peking University, Beijing International Center for Mathematical Research. And another affiliation is the Center for Quantitative Biology. So this center is for we have different people from math, from physics, from biology, from chemistry. So that's the Quantitative Biology Center. Today I'm going to talk about noise attenuation during the development of spatial pattern. Before my talk, I would like to introduce my center. So I believe not many of you visit us before, right? So how many people went Peking University before? Only a few, right? So yeah, that's our math center. We are in the north part of our Peking University, near the Weiming Lake. That's the most beautiful part in our campus. We have 7 Chinese classical courtyards. Okay, that's the picture for our different courtyards. My office is right here. So very beautiful place. Every year we organize conference, workshops, summer school. So and last year we built this PKU biomass lab. We have 12 junior faculties doing biomass. We cover most areas of biomass. So welcome everyone to visit us. Actually, we organize some summer activities this year, including a summer school and international conference in July. Okay, so that's my lab. Where I have 8 graduate students. My training background is applied and computational math. Originally, I do this phase field model in solid state nucleation stuff. Okay, computational materials science. Then I changed my research topic to computational system biology, mostly focus on developmental and cell biology, involves pattern formation, noise, stem cells, reprogramming, cell polarities, this kind of stuff. So today I'm going to tell you 2 simple but very interesting stories. I hope you can relax yourself, but please do not fall asleep. Okay. The first one is temporal noise attenuation in 3-node network. The other one is spatial noise attenuation during boundary sharpening. For the first one, actually, we are looking for the network topologies that can achieve dual functions, in particular adaptation and noise attenuation. That's the joint work with Tang Chao and my students Lingxia Qiao and Wei Zhao. We know noise is area in biological systems. In gene expression, there are lots of noise fluctuations, like this bacterial cells produce different amounts of proteins. It is the wind disk in morphine gradient. If you measure this gradient instead of the smooth gradient, actually that contains lots of noise. So one question we would like to address is how can biological systems adapt, change those fluctuations in environment? There are two questions. One is they need to maintain the mean. Okay, it means the environment has a sudden change, like the input signal. They can change like 20% or the 1-fold and 2-fold change. How the system can adapt such change? The other one is to reduce variance. It means there contains a lot of noise fluctuation. How to attenuate those fluctuations? To maintain the mean, we need adaptation. What's the adaptation? The input signal I1 change to I2 is could be like 20% change or 1-fold change. It doesn't matter. Okay. Then output is O1. They can sense such change. Okay. But eventually they can adapt to the close to the original level. In this case, how we quantify that? We define two quantities. One is sensitivity. It's O peak minus O1 divided by O1 over I2 minus I1 divided by I1. That's the sensitivity. And another one is the precision. So that's the O2, this guy, minus O1 divided by O1, then divided by the input difference. Okay, so a good adaptation means high sensitivity and high precision. Okay, and Tang Chao's lab, they searched 3 nodes network, all possible networks in this case. Okay, the totally there could be over 16,000 networks. Guess how many? Guess how many can have adaptation? It's about 395 networks. They can perform a good adaptation. There's all possibilities. Either positive feedback, negative feedback, activation, inhibition. Yeah. Okay. So among those 16,000 networks, there are 395 networks can achieve a good adaptation. Among those good adaptation networks, okay, we have the minimum network. That's the 3 nodes network. Okay, only has 3 links. Okay, you see here only have 3 links here. There are 2 types. One is the negative feedback buffer node loop. Okay, another one is the incoherent feed-forward loop. Okay, so a good adaptation network either contains one of them or contains both of them. Okay. How about the noise, the reduced variance? In that case, we need a good noise attenuation. It means the input has a large fluctuation, output small fluctuation. In that case, we can define this R2 is just the CV for the output divided by the CV for the input. Okay. In that case, the minimum network is 2 nodes, and actually there are huge literatures to discuss how to attenuate the noise, right? So, like for example, some papers claim the negative feedback loop can reduce noise. Some papers claim positive feedback can reduce noise, and also some papers show that the time scale actually matters. Okay, so all those kind of network discover the minimum network is 2 nodes. And here we are looking for the circuits capable of both adaptation and noise attenuation. It means the input signal has a sudden jump with a fluctuation. In that case, we can discuss the 3 nodes. We can start from the 3 nodes network. Okay. Also, we can extend to 4 nodes. How we proceed? So we can, based on the network topologies, it means we just search noise attenuation networks through all 395 networks that can perform adaptation. That's the easy job, right? So we just, among all those networks can perform adaptation, we just search among them how many networks can have good attenuation, noise attenuation. Then another one, you can, based on the time scale, you can change the time scale to achieve both functions. At the beginning, we thought it is an easy job. Okay, it is an easy job because we just do the random search. Okay, so the searching strategy is following. We use such kinetic equations, okay, describe those 3 nodes. Okay, this both A, B, C, that's the active form and the inactive form, and we use this kind of typical dynamics to describe it. And the searching criterion is adaptation, which means we have good sensitivity is greater than 1. We set a threshold. Precision, we set is greater than 10. Noise attenuation, it means the R2 is less than 0.2, but maintains the good sensitivity. Sensitivity is greater than 1. Okay, and both, it means we have this criterion. Okay, guess what we got? Actually, we searched all those 395 3 nodes adaptation networks, and each network we used 10,000 times simulations of different kinetic parameters for one network. Then that's the plot we got. We found actually we failed to achieve such dual function. Okay, here the horizontal axis is Q1. It means the numbers of simulation, has a good adaptation. Y-axis, vertical axis, Q2 denotes the number of simulations that have good noise attenuation. Actually, you see in this area, it's empty. Okay, it's very hard to use this random, just to give the random different kinetic parameters to find a good network to have both functions. Why is that? We try to dig out. We found 3-node networks, so trade-offs between adaptation and noise attenuation. That's the typical what happening. That's the input. Okay. In some case, we found the output has a good sensitivity, but they has bad noise attenuation. Some networks they have good noise attenuation, but bad adaptation. This is because we measured the relations between different quantities, sensitivity and the precision. Here you can see that that's the negative relations. Okay, so it means high sensitivity. It means bad precision. Okay, it's normal. And for the R2 and the sensitivity, here you see high sensitivity. It means bad noise attenuation. Okay, because R2 is large, it means it has bad noise attenuation. Okay. So what's next? So what's the solution for this trade-off? We add time scale. Okay. In this case, for those kinetic equations, we add different time scale for the A node, B node, and C node, and see if we use this different time scale, can we obtain those good dual function? We found the result is actually it helps. Okay, adjusting time scale can improve noise attenuation while maintaining adaptation in both incoherent feed-forward loop and the negative feedback buffer node loop. Okay, in that case, we see the different color means the different ratio for this tau C over tau B. Okay, and here we found this ratio. We increase this ratio, we have the larger sensitivity. Okay, what x-axis is sensitivity, y-axis is R2. Okay, and here along the same color, we found actually decreasing tau C. Okay, decreasing tau C while maintaining this constant actually can reduce R2. Okay, so that's a good strategy. That's the one simulation. Okay, in this simulation, we slow both node B and node C proportionally. We found is actually we got better. We can maintain this sensitivity, and also we have better noise attenuation. Okay, it seems this strategy works perfect, but no free lunch. Here we found that cost of changing the time scale actually it increased the adaptation time. Okay, here we do some quantitative analysis, but here you can see the picture just using this one. Okay, we decreasing tau C, it means we slow the node C, then it increase the response, the adaptation time. Okay, so you can think about in reality the system won't adapt quickly. Okay, you don't want the system response after a few cell cycles. It takes too much time. Okay. So, but that's what we found for the 3 nodes. Okay. So beyond 3 nodes network, okay, we just got preliminary results. Okay. We extended to the 4 nodes, 4-node networks, and we found this 4-node networks can do more. Okay, it can achieve dual functions without increasing adaptation time significantly. Here we have different strategies to construct 4-node network. Okay, so for example, we have different circuits. Okay, first we this input. Okay, we perform the adaptation circuit, then we perform the noise attenuation. Or you can perform the noise attenuation first, then let it adapt. Also, you can do both. Okay, one circuit it denoise, one circuit to adapt. Okay, that's the typical examples for the for these different strategies. And in this case, we found actually we let the input signal first we denoise first, then we let the signal adapt. Using this strategy, we got a good adaptation and a good noise attenuation without increasing adaptation time. Okay, this means, okay, so it's easy to understand because you think about in this circuit we just denoise, we do not reduce any sensitivity. So the high sensitivity signal got went into this adaptation circuit, then it can adapt eventually. So it's a good strategy to perform such dual function. Okay, and here we try to explore more to find out the other possibilities for the 4-node network and find the more analytical reasons to explain that. Okay, that's the first story, very simple. Second one is we extend to the spatial noise attenuation. Here we studied boundary sharpening in biological systems. Two systems. One is the zebrafish hindbrain, the other one is a mouse forebrain. Okay, that's a collaboration work with Qingye, my former postdoc mentor, Tom Schilling and Adam Nuki. They are also the biologists in UC Irvine. So in this case, we are trying to answer how cells know where they are. Okay, we try to in this case for the classical morphogen gradient, we know we have a morphogen gradient, then we can produce this French flag pattern. But there are many in the morphogen gradient that contains many noise, so that around the boundary actually they produce lots of noise. This noise, it means the cells appear. The cells have different phase, okay, because of the noise here. And how to sharp this boundary is a very interesting question to address. The first example we try is the zebrafish hindbrain. Okay, here that's during the development we found there are 7 segments for the zebrafish hindbrain, and they use genetic control. This cross-20 gene expressed in R3 and R5, those 2 regions. And in the middle, that should be R4. Okay. So Tom Schilling's lab, they did this boundary sharpening experiments. At the beginning, you see this cross-20 gene expression has lots of noise, and this noise becomes becomes less and less over time. Eventually, the boundary becomes sharp. Okay, after 2 hours. Now it's how it happens. Okay, because that's the zebrafish should have the normal function. They have to achieve such boundaries. Okay, to answer that, we build such network, the mathematical model to address that. Okay, there's RA produced in the posterior area. They diffuse from the gradient. Okay, then small molecular RA can move into the intracellular RA. Then we have CYP26. It's an enzyme to give the robust shape for the intracellular RA. And downstream we consider 2 genes. One is HOSP1, the other one is cross-20. They have both genes activated by the intracellular RA. They also have this auto-regulation and mutual inhibition to each other. Okay, so in this case, we use this reaction diffusion equations to describe morphogen equation for extracellular RA, intracellular RA, and then we use the ODE systems to describe HOSP1 gene expression and the cross-20 gene expressions. Okay, so that's the mathematical formulation. Without noise, we just think about the deterministic model. Okay, how to generate such alternative pattern. This R3, that's cross-20, HOSP1 cross-20, this alternative pattern. In this case, we found actually this initial level of HOSP1 and mutual inhibition are essential for this normal gene pattern. Okay, if the initial HOSP1 is too low or too high, it cannot produce such alternative pattern. Okay, only a certain range it can produce the normal gene pattern. And here we found experimental support, okay, to show this initial HOSP1 pattern in the chick hindbrain. Okay. Then we try to answer the question of the boundary sharpening. Actually, that contains two questions. First one is, during the segment development, what kind of noise induced the initial noisy boundary? Okay, that could be extracellular or intracellular noise, morphogene noise, gene expression noise. Okay, after that we can we try to answer how can this noisy boundary become sharp? It could be some regulation of morphogene, or it could be some type of noise. Our approach is, first we try to use some theoretical analysis. We use the idea of the real events to compute minimum action path to estimate gene switching probability. Then we use both stochastic PDE model, this continuous model, and we use discrete model, use SSAA to simulate the dynamics. Okay, but the key idea is this real event. Okay, let me use just a few slides to introduce a little bit what's the real event. Okay, so the real event is most complex dynamics actually proceed in the form of real events. In simple words, real events is just say it's a small probability jumping process. Okay, for the system jump from one metastable state to another metastable state driven by very small noise. Okay, so this kind of dynamics can use this following equations to describe. X dollar is equal to negative gradient U. U is just free energy. Okay, plus noise. Okay, this small noise can drive the system from one metastable state to another one. Okay, in that case, it has to jump over the energy barrier. Okay, and here find out the such energy barrier is a hard problem, because this energy barrier is a set point. It's an unstable solution for the equation. And most people use this transition state theory to describe it. Okay, it just says these transition states are the dynamic bottlenecks for the transition. With very high probability, transition paths have to pass through a very small neighborhood of this transition state. Once transition state is passed, then the system can relax to the new state by following its own intrinsic dynamics, okay, on its relaxation timescale. Okay. By using this random dynamic systems, we can use this probability described by this action functional. To minimize this action functional, give you the most probable path. It's defined like that. Okay, so with this most probable path, what we can solve, what question we can answer. Generally speaking, there are three classes of problems we use the real event to explain. The first one is A to B problem. A means the initial steady state. B is another steady state. Then we try to find the most probable transition path from connecting A to B. Okay. The other one is the exploration problem. It means we only know the initial state A. Then we need to go out to find out all possible outcomes. Third one is to accelerate dynamic simulations. It means like the protein folding. Okay, you have to fold many times to reach the final configuration. Okay, how to accelerate such dynamics. Okay, you don't want it trapped in a local minimum. Again, the computational difficulty of saddle point is this saddle point is unstable. It's much more challenging than finding a stable energy minimizer. Also, in high dimensional space, the energy landscape is often complex. It increases the difficulty of finding the saddle point. Okay, what we use this real event to explain the boundary sharpening. Here we calculate all possible states for this cross-20 house B1. Okay, then we try to find the minimum action path connecting different states. Okay, that's the technical works. Okay, don't worry. Okay, then we use the minimum action path to estimate the gene switching probability. Just use this picture to show that. Okay, here our idea is this the morphogene gradient and contains noise. Okay, for the morphogene, this high level morphogene gradient, they have one state, fixed fate For the low level, it has another fate. But in the middle, around the threshold, okay, here you can see that those two genes mixed together, two cells mixed together, but they have different stability. Okay, that's the key issue. In that case, if we know one state is less than the other state, in that case, if we contain a gene expression noise, this noise can drive this green one to switch to the red one. Here you think about if we can switch those two green cells to the red cells, then you have the sharp boundaries. Okay, that's the quantity we want to measure. Okay, is that so? We use the stochastic PDE to simulate that. First, we just consider the morphogene noise. In that case, because morphogene contains noise, we do observe such noisy pattern. But however, this noisy pattern cannot be sharp because we don't have induced any mechanism to reduce those noise. Here, in the second in the second movie, we add both morphogene noise and the gene expression noise. In that case, the morphogene noise induces the initial noisy pattern, and the gene expression noise actually can induce the gene switching from this blue one to the red one because of this gene expression noise. Here you see the later, this gene expression noise can induce those two blue cells to the red cells. Okay, then we got the final sharp boundaries. Okay, so here we found actually this intracellular gene expression noise plays some good role. It can induce the gene expression and coordinate cellular decisions leading to the boundary sharpening. Okay, so we measure the boundary sharpening. This experiments and simulation agree with each other, and from the in vivo experiments, we found that here those cells appear in the wrong place. Okay, those either host B1, cross-20, or co-expressed cells. Okay, at the early time, middle time, and later time. And here you see in the later time, the cells appear in the wrong place, become less and less. So it means that those cells switch to the right fit, the right face. Okay, and also we explore the gene expression noise amplitude frequency to see to find out what's the optimal ratio for that. Okay, so that's the example for the zebrafish hindbrain. Okay, you may wonder if this mechanism only works for this particular network, or it works for the other systems. Okay, here, lucky for us, we found this mouse forebrain also has this boundary refinement, this phenomenon. Okay, in that case, actually, we have both BMP FGF network. We have two gradients. Okay, so that's even better. Okay, so in that case, first we start with the single cell model. Okay, we build the dynamics for BMP and FGF intermediates. Okay, then we have the BMP FGF target genes using those Hill functions. From the experiments, actually we found those developmental delay. Okay, so experiments show that this FGF suspension of BMP target genes is slow due to translation, so that we can propose a delay model for the FGF intermediate by introducing a transient, this FS, okay, transient FGF component, okay, between B and F. Okay. So then we try to find this delay model, how delay model affects this temporal noise attenuation. Okay, here we found this noise from BMP and noise from FGF. We found that slower FGF actually can attenuate, can have better noise attenuation. And from experiments, Barrow and our simulation, they consistent with each other. So we found that the slow FGF enables noise attenuation for BMP target genes. How about spatial noise attenuation? Originally, we have noisy pattern like that. In the zebrafish hindbrain, we have single gradient RA. In that case, they can produce gene switching like that. That's one-way gene switching. Okay, this we can switch red to blue. Turns out it can switch those 4 red to blues. However, for the other side, we cannot switch because in those areas, both gene switching probability are low. Here, for the mouse forebrain, we have 2 cross gradient, BMP and FGF. Okay, this gives us a better gene switching probability. They can do 2-way gene switching. Okay, blue to red, red to blue. Red to blue and blue to red. In that case, okay, on the one side, those red gene can switch to blue. On the left-hand side, those blue cells can switch to the red. Okay, so in that case, we found actually 2 cross gradient improves the boundary sharpening than single gradient. Explanation from the transition state theory is just like that. Okay, we have the noisy pattern here, but they produce different metastability for the blue cell and the red cell on the different locations. Okay, if the FGF is high, it means the red cell is more stable. BMP is high, it means the blue one is more stable. Okay, so then this BMP FGF can induce 2-way gene switching, leading to the better boundary sharpening over time. Okay, so that's the spatial model. Okay, we add the noise. Here actually we use the bounding motion, but here this term doesn't matter. You can use the white noise, color noise. Okay, that doesn't matter. Here we test the boundary refinement 2 gradient slow FGF action. Okay, that's 2 targets. First one is the wild type. Okay, at the beginning you see the noisy pattern, then it becomes sharper and sharper. And here, second one, we use the fast-slow inhibition. Okay, one inhibition is fast, the other inhibition is slow. Here, you observe the developmental delay. Okay, that's the we observed from the experiments. We induced such fast-slow inhibition in the model. Actually, we found that this fast-slow inhibition results the better boundary sharpening than the fast inhibition. Okay, that's the 1 time is equal to 300. That's the Cauchy steady state. Okay, here you can clearly see that this fast-slow inhibition leads to the better boundary sharpening. Okay, here that's the critical role of FGF in boundary refinement. Okay, that's a wild type. Okay, the wild type BMP FGF. Okay, you got such boundary sharpening. If you decrease the FGF by 30%, then it just gives you the boundary shift, right? And here you see that this boundary refinement is less, it contains many noise here. Okay, here we induce slower FGF, it's 100 times slower. Okay, in that case we found there is a developmental delay. Okay, experiments. Okay, then it results in the better boundary refinement compared to the wild type. Okay, so and also we found this experimental support to see in vivo data to reveal temporal refinement for this cortex dorsal midline boundary refinement. Okay, that's the quantitative plot for this boundary gradient, sharper and sharper. Okay, so another experiment we did is the transient FGF perturbation. Okay, we inject those transient FGF perturbation, see if it can recover the original boundary sharpening. Here, that's the results for the transient reduced FGF with the fast FGF action, and the second one is the transient reduced FGF with slow FGF action. Here you see that the slow FGF action recovers better boundary sharpening than the fast FGF action. Okay. So here they just show the slow FGF action is the key to robust recovery after perturbation in spatial model. Okay. It is consistent with the experiments. Okay. In the experiments, they inject the transient perturbation to FGF signaling in vivo. Okay. Then this boundary a sharp boundary with noise attenuation reflects the developmental catch-up in both experiments and simulation. Okay, and we can see those noise in the FGF signal is well buffered. Okay, so that's the summary. I gave you two very simple stories. One is the three-node network topologies to achieve both adaptation and noise attenuation mechanism. The other one is we proposed a new noise attenuation mechanism for the boundary sharpening. Okay. That's my collaborator funding support. That's all. Thank you very much.
Questions? So can I ask a question? These nodes and these arrows you sort of left them abstract, I think probably deliberately, but do you know what they are, or can you give us like an example of?
That's a very good question. Actually, we do found the example because in three-node network, we found actually no single topology can achieve both adaptation and noise attenuation these two functions. Okay, then we extend to the four-node network. I didn't show here, but in reality, we found one. We try to find more, but at least here we found one in the real network. They do have this positive feedback at the beginning. In the upper level, they can reduce noise. Then they go to this adaptation network circuit to adapt. So that's a very good suggestion, and we are doing right now. Yeah.
First of all, why is there any reason to restrict to three-node networks? Because maybe you need like 5-7 variables to achieve this.
Yes.
And another, this time scale is not just changing the coefficients. So you say that you change time scale, one of them. It's not equivalent to other set of reaction rates.
Okay, so for the first question, why we started a three-node network, right? Three-node network is the minimum network to achieve adaptation. That's the starting point. And we know two-node network is the minimum network to achieve noise attenuation. So to study both functions, to do these dual functions, we think the minimum network should be we should start from the three-node network. So that's why we started that. And yes, if the three-node network can achieve both functions, that's perfect. Then it means three-node network is a good network to achieve many functions. But here we found actually you can use time scale to achieve both functions, but the price you paid is you need to increase the adaptation time. So we think in reality this strategy may not work. So we extend three-node network to the four-node network. We build some module to attenuate noise, some module for adaptation. We combine them together to achieve both functions. So that's the four-node network. The idea for build up this four-node network. Okay. The second question is the noise attenuation. The time scale, right? So yes, here the time scale we add is we use the simple way to do that here. Yes. Okay. So there are some coefficients, okay, corresponding to the strength for this either positive feedback or negative feedback. Okay, and here we add this tau A, tau B, tau C. It means we add this node time scale. You can easily to adjust. So actually, the tau A is always equal to 1. So we only have two new parameters you can adjust. So instead of you changing individual, actually we can change one to achieve. We can slow B or we can faster this node.
It's like you orient the search for reaction rates in different regions. If I understand, for each network like this, you make a search in the space of parameters.
Yes.
So you just orienting the search to different area by
Yeah.
changing the time scale.
That's right. Yeah, you can think about it as also is try to find the parameter, red parameters, right? Red reaction rates to find the good ones, the optimal ones, right? So here, I think adding those the tau b, tau c is you can change those parameters proportionally. So it's an easy way to search. Yeah, you said it definitely correct. Mm-hmm.
And are you doing the like a full search in the four-node the networks? Because it's possible that you find a new solution. You show the examples where you combine the existing solutions for adaptation. But it's possible that like you find a completely new topology where it combines the adaptation and denoising in a novel way.
Here, for the four nodes, because for the adaptation, we know the minimum network. Either it belongs to the negative feedback buffer node, or either belong to this kind of incoherent feed-forward loop. Okay, and for the noise attenuation, we know this kind of positive feedback can attenuate noise. So that's the basal. That's the minimum module for the adaptation. That's the minimum module for noise attenuation. For the four-node network, we try to combine them together. See, attenuation first, or adaptation first. So you have different strategies to combine them. You result different output. You got the different outputs here.
Is it possible that if you don't use the solutions you found and you just search on a four-node network, you might find a new four-node version of the network that will do both at the same time?
Yeah, that's a very good suggestion. Here, we didn't do that because you see in three-node network, only for adaptation, all possible networks is already 16,000. Four nodes is increased exponentially. So it's hard to use a good just a random search. So here, that's why we use different smallest network. We combine them together. But actually, combine them. Each module you combine them, or you can just construct some tricky four nodes. This another story we need to explore. Yeah, because this combine them one first function A first, function B second, maybe not the only way to do that. So yeah, it's a good question to explore the different strategies for the four nodes network. Mm-hmm.
Okay, before we thank our speaker again, I'll tell you the schedule. So we have a half an hour break, and then at 4 we have another talk, and I'll ring the bell before the 4 o'clock starts. Okay, thank you, Lei, again.
Thank you.